Hello everyone, myself Dr. Parth Goswami and hope this video will find you all in a good health. Today I am going to teach you one another topic from immunology, that is a type 4 hypersensitivity reaction. This particular hypersensitivity reaction is a T cell mediated hypersensitivity, right? And the granuloma formation, the granulomatous inflammation is one of the classical example of type 4 hypersensitivity reaction. So let's begin with definition. So as we have discussed, type 4 hypersensitivity reaction is a T cell mediated hypersensitivity. Now the T lymphocyte could be of two type, CD4 or CD8 T lymphocyte. So the hypersensitivity could be of two type, CD4 T lymphocyte mediated inflammation, or it could be CD8 mediated cytotoxicity. So both the T lymphocyte can lead to type 4 hypersensitivity reaction. And this particular hypersensitivity reaction will occur in previously sensitized person, right? The person will develop a response to the antigenic exposure in a previously sensitized person. And so that the reaction will occur 48 to 72 hours after the antigen exposure, and that's why it is also known by the name delayed hypersensitivity reaction, right? All right. Now if you have not seen my other lectures of immunology, then see it before seeing this video, right? It will be easy to understand this hypersensitivity if you will see my video on T lymphocyte. All right. So first we will begin with type 4 hypersensitivity reaction that is mediated by CD4 T lymphocyte. Alright. So here the antigen exposure could be of exogenous from the environment, or it could be endogenous self cell, right? It could be endogenous, means the body will develop response against the own cell antigen, which is known by the name autoimmune disease. Or it could be the reaction against the environmental agents like bacteria, virus, etc. Like that. Okay. Now, in this particular hypersensitivity reaction, the CD4 T lymphocyte will get activated, right? And they will produce a certain cytokines like that of interleukin 1, interferon gamma, right? Interleukin 17, and these cytokines is responsible for inflammation and tissue damage. So the person will have a tissue damage and subsequently symptoms, right? In that way it will act. And in this hypersensitivity, now if you remember my lecture of T lymphocyte, then CD4 T lymphocyte, which is also known by the name T helper lymphocyte, could be of three variety: Th1, Th2, and Th17, right? And this type 4 hypersensitivity reaction, which is mediated by CD4 T lymphocyte, will be because of two T helper cell, either Th1 or Th17. So I want to say that in this hypersensitivity, the T cell can be differentiated either into Th1 or into Th17. All right. Now this particular diagram demonstrates that how CD4 T lymphocyte lead to hypersensitivity reaction. So if you observe carefully that then here there is a presence of bacterial antigen, right? This is the antigen. All right. Now this antigen will be carried out to the T lymphocyte, particularly naive T lymphocyte, right? The T lymphocyte initially would be of immunologically inexperienced, which is known by the name naive T lymphocyte, right? So this particular antigen will be presented to the T lymphocyte by the antigen presenting cell, the dendritic cell, Langerhans cell, macrophage, B lymphocyte, etc. are the antigen presenting cell. So they will present the antigen to the naive CD4 T lymphocyte. And now this CD4 T lymphocyte T helper cell can be differentiated either into Th1 cell, that is T helper 1, or T helper 17 CD4 T lymphocyte. Right, this is the subtype of CD4 T lymphocyte, the T helper cell. The CD4 T lymphocyte can be differentiated into Th1 or Th17. Now here, the Th1 T lymphocyte, if the T lymphocyte differentiated into Th1, then they will secrete interferon gamma and the tumor necrosis factor, which will activate the macrophage. And because of macrophage activation, there will be increased microbicidal power of macrophage, and they will lead to destruction of intracellular microbes. And meanwhile, granuloma formation can occur, right? And the macrophage contain many enzymes, many cytokines like that of interleukin 1, TNF, right? All that can lead to tissue damage as well. So there will be hypersensitivity reaction because of tissue damage. Now, if the CD4 T lymphocyte differentiated into Th17 T lymphocyte, then they will secrete cytokine number interleukin 17 and 22, which will do the function of neutrophil accumulation. They will recruit the more and more neutrophils, and you know very well that neutrophil contain uh, hydrolase, right? It will contain lysosomes, right? They contain many hydrolytic enzymes, so they can do the tissue damage, right? So because of macrophage activation and because of neutrophil recruitment, there could be tissue damage in this particular hypersensitivity reaction. All right, hope you understand this diagram well, right? So I will proceed further. So basically, there are three main example of CD4 T lymphocyte mediated inflammatory reaction, right? This particular reaction could be of three variety. There are three classical example. The one is tuberculin test, then chronic granulomatous inflammation and contact dermatitis, among which this granulomatous inflammation is most important, right? It's a favorite topic of every examiner. So let's begin in the detail. These all three examples of CD4 mediated type 4 hypersensitivity. So first one is tuberculin reaction, that is MONTOUX test. Now this particular test was done in the past for the tuberculosis diagnosis, right? Nowadays this test is not used much. So what is tuberculin reaction? The first of all, the tubercle bacilli, right? The tubercle bacilli and the protein that contain the tubercle bacilli, which is called as a tuberculin, or we can say purified protein derivative, will be injected in a person intracutaneously as an injection, right? It will be injected subcutaneously. Now if the person is sensitized previously, right, we can have idea that whether person sensitized to tubercle bacilli or not. So if the person sensitized, if the person has exposure to tubercle antigen, then the injection site will become red and indurated immediately within 8 to 12 hours. It will reach maximum in the 24 to 72 hours, and gradually after 72 hours, after 3 days, it will slowly subside. So if this happen, then it means that person is sensitized previously to the tubercle antigen, and so tuberculin test is positive, right? All right, so we can say that whether person has immunity to TB bacilli or not. And if you observe microscopically such red and indurated lesion, then obviously it is CD4 mediated inflammation. So the person will have perivascular coughing of CD4 T lymphocyte, right? The macrophage could also be there. All right. And now we will see chronic granulomatous inflammation. The second example is granulomatous inflammation, right? See friends, this chronic granulomatous inflammation is a special type of chronic inflammation, right? It is a special variety of chronic inflammation, right? And the tuberculosis and the leprosy are the classical example of it. There are many examples of granulomatous inflammation, but today we will take tuberculosis as a prototypic example, right? It is also type 4 hypersensitivity. Let's understand it in detail. It's very easy to understand, right? And it's a most important topic in the pathology. So let's begin the granuloma formation mechanism, granulomatous inflammation. So first of all, here you have the bacterial antigen, particularly tubercle bacilli, right? This is the tubercle bacilli. Now this tubercle bacilli will be engulfed by any of the antigen presenting cell, and the antigen presenting cell will present these tubercle bacilli to the naive CD4 T lymphocyte. It will present the antigen to the T lymphocyte, right? So as we have seen, the T lymphocyte can be differentiated into Th1 or Th17. Here it will differentiate into Th1. Because of differentiation into Th1, they will secrete interferon gamma. We have seen it previously, right? Th1 will secrete interferon gamma, and this interferon gamma will activate the macrophage, right? We need to kill the tubercle bacilli, right? The tubercle bacilli need to be killed. So the macrophage are activated. They are the powerful phagocytic cells, so that they can kill the tubercle bacilli, right? So the macrophage will get activated. And see friends, this is the magic, right? This is the magic. See, the normal macrophage is having the eccentric nucleus and foamy cytoplasm, right? This is the normal macrophage. But whenever they are activated, they will get converted into epithelioid cell. What do we mean by epithelioid cell? Here the macrophage will change its shape, right? The shape will change. See, it will get converted into elongated spindle shaped cell with the indentation here, right? So it looks like an sole of our shoes, right? It look like an boot shape or a sole of our shoes, right? It is slippery shape, which is known by the name epithelioid cell. So here the macrophage will get converted into epithelioid cell, right? Elongated. Here you can see that this is a slippery shape epithelioid cell. Now friends, when this epithelioid cell, right? This is the epithelioid cell with central nucleus, right? These are the epithelioid cell. Now when these epithelioid cell fuse together, here they will fuse together, right? These all epithelioid cell can get fused together, right? Their cytoplasm can fuse. So whenever they get fused, these 5 to 6 epithelioid cell will form a giant cell, right? Whenever they will fuse, they will form a giant cell. Giant cell means there are presence of many nucleus, right? There are presence of many nucleus within the cytoplasm. So when epithelioid cell fuse together, they will form a giant cell, right? And if the nucleus arrangement is horseshoe shape, right? Here you can see that nucleus arranged in a horseshoe shape, then it is known by the name Langhans giant cell, right? So the person will have giant cell formation in granulomatous inflammation. Obviously this is mediated by lymphocyte, right? So there will also be presence of CD4 lymphocyte. These blue dots are lymphocyte, right? These blue dots are lymphocyte. All right. Macrophage can also be present, and as it is chronic inflammation, there will be presence of fibroblast. And here, because of cell death, there could be presence of special type of necrosis, which is caseous necrosis, right? The person can have caseous necrosis as well. So let's summarize here. Because of macrophage activation, the person will have epithelioid cell, right, which is a modified macrophage. When these epithelioid cell fuse together, they will form a multinucleated giant cell, right? Because of chronic inflammation, the person will have fibrosis. There will be fibroblast. And as it is type 4 hypersensitivity, which is mediated by lymphocyte, so obviously there will be presence of lymphocyte. And additionally there will be presence of caseous necrosis. So all these 5 components, caseous necrosis, lymphocyte, fibroblast, epithelioid cell and giant cell, together will form a round structure which is known by the name granuloma formation. So there will be granuloma formation in the special variety, and that's why it is known by the name chronic granulomatous inflammation. All right. If you see this diagram carefully, this is a histopathological image of chronic granulomatous inflammation in which there is a presence of many granuloma. These are the granuloma. See it. See, follow the laser light. These are the granuloma, right? These all are the granuloma. If you see it carefully, this pink color structure is caseous necrosis. These blue colored dots are lymphocyte, right? There is a presence of fibrous septa, and there is a presence of multinucleated giant cell as well, right? So all this structure will form a granuloma, which is known by the name chronic granulomatous inflammation. So in the chronic granulomatous inflammation, the person will have granuloma formation, and this is the structure of granuloma, which is a most important short note, or we can say MCQ, right? This is again the granuloma, and here there is a presence of giant cell. These all are giant cell, right? All right. Third example of CD4 mediated hypersensitivity is contact dermatitis. So certain environmental agents like that of IV poison, nickel, chromium, certain dyes, cosmetic or soap, whenever the person will be in contact of this substance, it can evoke a inflammation in the skin. So in the skin there can be formation of blister with the redness, right? Which is known by the name contact dermatitis. Here the lesion is present in the skin, that's why it is known by the name contact dermatitis. All right, so this was all about the CD4 T lymphocyte mediated type 4 hypersensitivity. Now we will see the CD8 T lymphocyte mediated cytotoxicity, right? The CD4 explanation is over. Now I will explain CD4 T lymphocyte mediated type 4 hypersensitivity. Here what happened, friends, whenever the whenever the antigen exposure is there, the antigen bearing target cell will be directly killed by the CD8 T lymphocyte, right? They will directly kill the infected cell, and in such way they will eliminate the infection. The examples are type 1 diabetes, graft rejection, viral hepatitis, right? Tumor cells, right? In the neoplasm. So in all this variety, the type 4 hypersensitivity will be mediated by CD8 T lymphocyte, and they will directly kill the infected cell. There are two ways of killing the infected cell by CD8 T lymphocyte, either perforin granzyme system or Fas ligand Fas pathway, in which apoptosis will occur. I will not explain both these mechanism in the detail because I have already mentioned this. I will already mention the details of this way of killing in my T lymphocyte lecture, right? So if you have not seen it until now, then you can check the playlist and see my lecture on T lymphocyte, right? CD8 T lymphocyte can kill the infected cell by two way: perforin granzyme system and Fas ligand Fas apoptosis system, right? All right. See, these are the another example of type 4 hypersensitivity reaction. The first example is rheumatoid arthritis, right? In which the person will have a patient will have chronic arthritis with inflammation, and there will be destruction of articular cartilage, right? And the antigen could be of collagen or citrulinated cell protein, right? It's autoimmune disease. So the first example is rheumatoid arthritis. Second example is multiple sclerosis, right? Here the antigen is protein in the myelin. The hypersensitivity will be against the protein antigen in the myelin, right? And so there will be demyelination of central nervous system with the inflammation, and the person can have paralysis or optic neuritis, right? Third example is type 1 diabetes, in which the hypersensitivity will be against the pancreatic islet beta cell, right? It will act as an antigen. And so the person can have chronic inflammation in the islet cell of beta cell, and person can have diabetes, right? The fourth example is inflammatory bowel disease. It's again autoimmune disease in which hypersensitivity could be against the bacteria or self antigen, right? Yeah, there will be chronic intestinal inflammation, so the person can have intestinal obstruction, vomiting, etc., like that of complaint, right? The fifth example is psoriasis, in which the antigen is unknown and there will be destructive plaque in the skin. It's a skin disease, right? Autoimmune psoriasis. And the final example is contact sensitivity, right? In which various environmental chemicals can act as an antigen, and the person will have dermal inflammation, right? There could be epidermal necrosis as well. Skin, there could be necrosis in the skin, right? So these are the example of type 4 hypersensitivity reaction, right? tuberculosis, leprosy, right? And these are the additional examples, some other examples: rheumatoid arthritis, multiple sclerosis, type 1 diabetes, inflammatory bowel disease, psoriasis, and the contact sensitivity, right? Among all these examples, the most important example of type 4 hypersensitivity reaction is chronic granulomatous inflammation. Tuberculosis, leprosy are the two main example of chronic granulomatous inflammation, right? You have to write down this example first. So this is the chronic granulomatous inflammation. All right, it could be mediated by CD4. The type 4 hypersensitivity could be mediated by CD4 or CD8 T lymphocyte. Let's see some of the golden points, right? If the person is immunosuppressed, right? If the patient is diabetic, if the patient is taking chemotherapeutic drugs, then tuberculin test could be negative, although the person sensitized to tubercle bacilli, right? Interleukin 2 can be secreted from Th1. So, when the macrophages are activated by interferon gamma, which are the characteristic feature that will develop in the macrophage, right? Whenever the interferon gamma activate the macrophage, they will develop certain characteristic feature. They will, whenever they are activated, the phagocytic power and the microbicidal, the killing of bacteria power, right? Bacterial killing power will be increased. They will express more and more MHC class 2 molecule on their surface, and we know very well that macrophage is one of the antigen presenting cell. So whenever there will be more MHC class 2 molecule on the surface, they can facilitate more antigen presentation, right? They can do more antigen presentation function. The macrophage can secrete interleukin 1, TNF, and chemokines, which can elicit inflammation. And they can produce interleukin 12 as well, which can again activate Th1 to secrete more and more interferon gamma, right? To activate the macrophage. All right. So this is all about the type 4 hypersensitivity reaction. If you have any doubt, then you can ask in the comment section. Till then, take care and see you soon in the next video. Till then, take care and bye bye. Yeah, thank you very much.